Hello and welcome everyone to ASBMB Live. I'm Raj Mukhopadhyay, the Chief Science Correspondent for the American Society for Biochemistry and Molecular Biology.
And I'm Jeff Hunt. I am ASBMB's Public Outreach Coordinator.
So today we have a great panel together. We're going to be talking about how a rare pediatric brain tumor has given cancer researchers new insight into mechanisms that take place in various cancers. The cancer we'll be talking about is called diffuse intrinsic pontine glioma, or DIPG for short. About 250 children each year get afflicted by this disease. There is no cure.
But there has been a new surge of hope lately thanks to a combination of clinical doctors, medical researchers, and patient advocates, and we are very lucky to have a lot of them on this hangout with us right now. So I'm going to have them introduce themselves one by one and give you a little background about where they're coming from. So Denise, let's start with you. If you want to unmute yourself.
My name is Denise Downing. I'm co-founder of the Children's Brain Tumor Family Foundation. My daughter, Caitlin, participated with Dr. Soydan in a clinical trial shortly after she was diagnosed with DIPG, and she passed away in 2012.
All right, Mark Curran.
Yes. I'm Mark Curran. I guess I'm hearing a bit of it. I'll try anyways. Cancer.
All right, getting a little bit of feedback. I'm not sure how that. Mark, do you want to go ahead? Mark, can you hear us?
I can hear you now.
We'll go to Michelle. Denise, can you mute yourself?
I'm sorry?
Can you mute yourself?
Sure.
All right, Michelle?
Yes.
There we go.
Hi.
There you go.
Hi, I'm Michelle Mange. I'm from Stanford University, and I'm a pediatric neuro-oncologist and a DIPG researcher.
Great. Nada?
Hi, I'm Nana Jabado from McGill University. I'm a pediatric neuro-oncologist and an oncologist without neuro, and I treat tumor in 2018.
Great. And, Oren, come back.
Hey,
Oren?
could you?
You're muted, Oren.
Oren, can you unmute yourself? Nope, we still can't hear you.
Oren, you're muted. Well, I can do the introduction for those who couldn't do it. So Oren Becker is a pediatric oncologist over at Duke University, and we had Mark Curran from Dana-Farber. I think Mark Soydan didn't get a chance to introduce himself, so if Mark Soydan can introduce himself.
Sure. I'm Mark Soydan. I'm the Director of Pediatric Neurosurgery at Weill Cornell Medical College in New York City and Memorial Sloan Kettering Cancer Center, and an avid investigator with regard to DIPG, and honored to share this panel with my colleagues.
And also in the audience we have Keith Deserich. He's the founder of The Cure Starts Now. We've been having technical issues in trying to get Keith on this hangout, but he is in the audience right now, and he will be willing to answer questions.
Cool. So in terms of questions, for those of you watching, you can post them on our Google Plus page. You can actually post them on this video Q&A feature that you can utilize, or you can tweet us at ASBMB, and we will do our best to get them answered. We're just going to start off with a little bit of background. I see we've already had some questions come in. That's great. And so anything you want to hear from the panelists, and hopefully we will get them all, and hopefully their microphones will be working and the cameras will be working and all that. So let's get started.
So before we jump in and start talking a bit more detail, why don't we talk a little bit about what DIPG is and what has been the research in the last 5 years that has made DIPG come into the forefront of cancer research. So shall we ask, let's get Michelle to describe what DIPG is, and then everyone can then start coming in with their different perspectives of what DIPG research has done in the last several years.
Sure. So DIPG stands for diffuse intrinsic pontine glioma, and it is a tumor that arises in a part of the brainstem called the pons. This is a critical structure for many forms of neurological function. So it's a very unfortunate location. It's a disease that arises chiefly in children, typically in mid-childhood, with a peak incidence of about 6.5. It's a disease that we are, as oncologists, very bad at treating. We made a little bit of progress about 35 years ago with the introduction of radiotherapy, which extended median survival by a couple of months. But since that time, despite great efforts, have made almost no progress in treating this disease effectively. And unfortunately, the median survival is only 9 months. The 5-year survival is less than 1%. So this is a generally uniformly fatal disease, which affects 200 to 300 children in the United States every year.
The two Marks, Nada, Oren, could you please now start talking a little bit more about what the research into DIPG in the last several years has started to reveal?
Whoever wants to. Nada, you're muted.
Nada, to unmute yourself.
So I unmuted. So I'll start because obviously Oren cannot seem to unmute. 5 years ago, DIPG was considered to be completely distinct as a tumor from other type of brain tumor. Several reasons. First of all, the location, and it's really something that's not accessible to surgery. And even people do not biopsy or take a small little piece to make the diagnosis really on imaging and imaging the way it looks like. And because it's such a very important structure, by doing a biopsy, people were afraid of creating more harm, and we didn't need it because the treatment was the same thing, and the patients thought there would be a fatal outcome within the 2 years following. We were very lucky. Within the first year, the vast majority of patients actually died. So on small biopsy and on autopsy sample, my group and the group of Susie Baker made 3 years ago in 2012 a major breakthrough. We actually uncovered an association of mutation in one per se that changes in the very conserved gene, something that is one of our keepers, the keeper of the integrity of our genome, a histone gene. There was this mutation that changes lysine to methionine in position 27 in 2 or 3 genes for the same type of histones that is present in the vast majority of tumors. This mutation seems to be associated with other mutations and may be responsible for this disease, for how it comes. It actually explains why this disease is so intractable, because it's actually what protects our genetic information and what makes it express and not express that is very sick. And the breakthrough was, okay, now we know what it is. Now the major issue is how to tackle this and how to treat this problem that now we know is there. This is what is potentially driving this tumor. I can't hear you.
Sorry, we were muted ourselves. Mark, either Mark, do you have anything to add to what Nada and Michelle have said so far?
Definitely reached the point now where, in addition to autopsy samples, we're actually able to biopsy the tumors at the time of diagnosis, which obviously is important because as we recognize the different mutations in these tumors, we can begin to think about individualizing the therapy based on the abnormalities of your specific tumor. And there's an international trial, or a large US trial going on right now, and another one in Europe where people are actually doing that. So it's not just theoretical, it's actual. We can safely biopsy the pons and get a small piece of tissue, and based on the advances in molecular biology, we can actually analyze them to tell us what's happening early and see if we can adapt therapy to improve treatment.
I'm actually going to now change the question a little bit because both Nada and Mark have mentioned things that I think Dr. Mark Swaidane can actually talk about, is the importance of being able to biopsy and get your hands on autopsy samples. How important has this been to the field?
It's been key. If we didn't have those biopsy and autopsy samples, First of all, we wouldn't be able to know what is it. People were treating it. There's more than 100 or 200 clinical trials with targeted therapy, with chemotherapy, with whatever therapy you want, and all of them have dismally failed. Before we biopsied these tumors or had access to autopsy sample, we didn't know about the histone mutation. Nobody would have guessed that this mutation was there and was present. So this is a major breakthrough. And I'm adding more to what Mark is saying. This is having a biopsy, a diagnosis is something that has to become more and more frequent. Why? Because, as I said, this mutation is associated with other things. We can't yet target completely this first mutation, but the other thing that it is associated with, some of them might be targetable today and can buy even more time and quality time to patients.
The availability of biopsy and autopsy samples has also led to the generation of cell cultures of DIPG tumors and correlate mouse models where we can put the tumor cells into the pons of mice and study the efficacy of a drug, for example, at least in a mouse model, and study the fundamental biology of the disease. So that has been an enormous help in trying to understand the most important functional targets and potential new therapies. So it's only through tissue donation. It has largely been at the time of autopsy, but is now increasingly, as biopsies are being done, tissue from the time of biopsies are being used to generate these cell cultures, and they're just an invaluable resource.
Dr. Mark Swaden, do you have anything to add?
Sure. I mean, from the standpoint of biopsy, it's, the brainstem has always been relegated as a surgical no-go zone to some degree. There's been two pressing issues that have pushed against the idea behind taking samples from these children. One has been philosophical, the other technical. I think on both of those fronts, certainly on the philosophical, we quieted those concerns based on the propagation of really amazing scientific invigoration that's happened mostly at the hands of most of the individuals on this panel today. This particular tumor is probably better characterized than any other central nervous system tumor in children in a very short time period with access to that type of tissue that we're talking about. And from the technical standpoint, it hasn't been an issue, and there is, of course, risk in any surgery. But I tend to think that it's always overemphasized and overestimated with regard to what that potential is, given that historical no-go zone. The last thing from a surgical perspective that I'll say, as much as there's been a call to sample these individuals at diagnosis or after treatment, or maybe longitudinally for that matter, one needs to be somewhat concerned about that universal call, and that is a technique and a technical aspect that does require some demand. It does require some surgical expertise. So while all of us would advocate that being done, all of us would also caution that it don't fall in the hands of every neurosurgeon who doesn't have a certain talent or technical background to be doing these types of biopsies in these children who are pretty sick.
We have audience questions coming in, but for now, we're just going to continue a little bit more to understand a little bit more about what the fundamental research is showing us about what's known about DIPG, and then we'll get to the audience questions. So, Nada mentioned the mutations in histones. Have there been discoveries of other types of mutations in DIPG that tell us a little bit more about what is happening in this particular type of cancer, and does it help us understand other types of cancers, be it pediatric or adult? And Oren, if you can unmute yourself and participate, we'd love to hear your thoughts.
No, we still can't hear
you for some reason.
Just see your face.
Yeah, so Nada, Michelle, Dr. Mark Swaden, or Mark Kieran, if anybody want to tackle that question.
So the mutation by itself is something that's quite unique to pediatrics. So the histone mutation are not very frequent in cancer when the gene is mutated by itself. However, the residues are getting hit by other ways in other type of cancer. So whatever we find, what it means, you have a gene and it gets changed to perform a specific task. The gene is hit by itself in those cancers. The way it changes is hit in other cancers, and it comes to almost the same effect at the end. So whatever we learn about histone and their regulation will be applicable to very high-grade other cancers. Okay? So this is to answer one part of your question. What we've learned also is that this mutation is present from beginning to end of this. We find it each time, and it's always there. So it seems there's a lot of debate as whether it's needed to initiate. Probably it is initiated, but is it needed to maintain? We don't even know that. But what we can only say for now is that whatever time of the disease, this histone mutant is present throughout, and this is there always. And now we have a little bit of proof to show that it is the first mutation that is happening in some cases. Is it throughout? We don't know. In this cancer, it is always associated with other mutations. And those mutations, it's very faithful. It has some partners that it loves. And when it finds a partner, it will be in the same patient throughout. So there are 2 or 3 mutations that are going to be the same in this specific patient. But the complexity on top of that, in order to grow, in order to be a little bit more aggressive, it's going to acquire other alterations. And those are the ones that we can tackle today. But they are not going to be the same throughout the tumor. I don't know if it makes it difficult. That means that if you look at the vast landscape, you'll have building in one area, you'll have nothing in another area, and you'll have a small little house. So it's very heterogeneous, which makes it a little bit even harder for targeted therapy. If we had a treatment that tackled this histone and we had proof enough that it is the one that is maintaining the disease, then it's going to be a big win. But for now, there is no proof that this is what's maintaining the disease, even if we're finding it throughout, and we have nothing yet to tackle it.
So does anybody else have anything to add on to what Nada has said?
Yeah, so I would just add that the understanding of the histone biology has led to some promising preclinical work using epigenetic modifying agents that in the preclinical models of DIPG that we mentioned earlier depended upon those tissue donations at biopsy and autopsy. There's at least evidence in mouse models that epigenetic modifying agents, there's a couple of different classes that have been shown in the literature to be efficacious, may be promising therapies, and there are trials coming up soon using those agents.
I think it's important to highlight and expand upon what Michelle was saying. The exploration of this tumor and the understanding of the molecular features driving these types of tumors doesn't necessarily equilibrate immediately with therapeutic ability, right? So there's still that fairly large jump between the molecular identification and the ability to kind of target and repair or modify these types of problems. So there is a large area of intensive research trying to focus on those specific mutations, but just because of the understanding is so well known doesn't necessarily mean that we have therapeutic agents that are designed today and available to be used immediately. So.
What I meant, there's nothing out there. What I meant, it's just that we need to better understand what is it that is driving, this mutation is driving, and those epigenetic modifier that Michelle and others uncovered and seem to be very promising in mouse model. Will they be enough, and what else is needed on top of them?
So I think something that's really interesting talking about going forward is how we've even got to this point, especially in terms of the sort of support and advocacy that's been done on behalf of this disease. So I'd be interested to hear people's thoughts about, how did this become something that was, quote unquote, a hot research topic, and how is it going to move forward in terms of getting support and getting more people working on these kinds of very rare diseases that don't have the major, let's say, sexy impact that of a disease that's been around for 30, 40, 50 years. And I think Denise can probably speak well to this about the foundation that she started and how that's impacted the research.
And Keith, who's also watching, can also jump in, and I'm sure there are other patient advocates out there who are listening who can also bring in their perspectives.
It took us a little while after we had died to decide
We're getting some feedback from you. Sorry.
Maybe turn down the volume of your computer, maybe that will help.
Is that better?
Much better.
Much better.
Okay, sorry. It took a little while after Caitlin had died before we decided to move forward with anything formal as far as our support of funding for research. We collaborated with some families that had worked with Dr. Sweden and Dr. Greenfield and the Children's Brain Tumor Project in New York, and put together a foundation and a nonprofit that would help to fund research for rare pediatric brain tumors and the work that those gentlemen are doing.
She's being modest, if I can. I think the success story is yes, what the science has produced. But the success story here is what the grassroots organizations, the individuals who have personal members invested in this horrible disease, driving this agenda forward. There have been many, many forces to push away or counteract an interest in doing this research. There's no honor. This is a horrible disease to face a family and a parent with. So there's that emotional, if you will, want to avoid and omit that from one's personal agenda, clinical or research. And then the other has been the funding. And I think there have been amazing resources generated through some of these foundations that have upstarted these type of collaborative efforts that you see based on this panel and beyond that here. These individuals would have never come together. And we are intimately involved every day of the week with doing research with each and every one of these. And these types of collaboratives would not have occurred in the void of that type of potential funding. So I think Denise is being extremely I should say, emphasizing the honor and really the emphasis that foundations have put on this. That is really, I think, one of the greatest success stories of this whole uprising, if you will.
Yeah, if I can add, I would add into that it's been remarkable that the National Institutes of Health, the American Cancer Society, the places where most research happens, particularly as funding has gotten tight, is the recognition that, with the focus on breast and prostate, colon, and lung cancer in adults, a rare pediatric tumor of 300 just doesn't hit anyone's radar, and that had the foundations and families not stood up to do this, we would still be in the dark today. So it's not just a question of they did it because they needed to. They really stepped into a place that other agencies were unwilling to do. And for any other rare disease, for any other circumstance, sometimes you rely on parents to take kind of destiny in their own hand. And admittedly, as you've already heard from Denise, these are families who've already lost their children. They're not doing it for themselves anymore, but they are hoping that they can prevent another family from going through this. And when you kind of think of what that means in terms of both who they are and what they're doing, it really is, phenomenal. That, none of this happens without them.
Yeah, I would just echo all of these sentiments and say that the progress that we've made in the last 5 years in the field is directly because of the efforts of parent advocates that are doing this for somebody else's child. It's really incredible. And in addition to the funding, which is so critical to support the research, the tissue resources are also a result of parents and families wanting to see that another child doesn't have to go through the same thing that they did. It's really been an amazing thing to watch and be part of.
So we had Keith Desserich, who unfortunately for technical reasons could not join the Hangout, but he is commenting on our event page. And he does say that, this is an opportunity. In many ways, DIPG forces us to think in a manner that is more collaborative and forces us to avoid duplicative funding. So while it's rare, the weaknesses can take us in a different direction, is what he says. And he also points out there is a limit of money for research, and it's just not enough. So he just points out that really the need to pool resources, especially money, to make sure that research into diseases like DIPG do get funded.
Cool. So we are going to take a question from the audience, and we have two, and it's sort of they're both sort of related in terms of drug treatments for the disease. So I'm going to try to summarize the two of them and bring them all together. Jesse Schumacher asks he says that one of the concerns about HDAC inhibitors, which you guys can explain HDAC inhibition, is the systemic toxicity from the amount that you're getting. And are there other options for delivering drugs to treat this disease? And has there been any sort of combination therapy that's been investigated? I know that's sort of a long question, but hopefully I made sense of it.
Sure, maybe I can start with that, and then I think probably Mark Suedwain could follow on my comments about some of his innovative surgical techniques. So HDAC inhibition is, HDAC stands for histone deacetylase inhibitor, and it's a class of drugs that has been shown in preclinical models to have some effect on DIPG growth. And the mechanisms of that are actually really interesting, and maybe we won't go completely into today, but they do seem to help to normalize the epigenome and to help to normalize gene expression in DIPG tumor cells. So HDAC inhibitors, many of them don't reach the brain very well, and there's one that we believe, based on preclinical work, to penetrate the brain a little bit. The question absolutely is can we give a high enough dose that enough gets in that we effectively treat the tumor? And the answer to that is that we don't know. That's the reason to begin to do the clinical trial. As we continue to gain more insight as to what drugs could potentially be combined and what drugs could potentially, or different strategies to potentially deliver this drug. HDAC inhibitors, as far as chemotherapeutic agents go, are not the worst actors. So unfortunately, the medicines that we give to fight cancer have a lot of unfortunate toxicities. They are toxins, they are poisons, they are designed to kill cells, and they are designed to kill cancer cells a little bit better than they, or significantly better than they kill normal cells, but there are always side effects of our medicines. Compared to traditional chemotherapy drugs, HDAC inhibitors have a relatively favorable side effect profile. Certainly there are important side effects, but they are not the most toxic class of drugs that we give to children with brain tumors, for sure. In terms of agents to potentially combine with HDAC inhibition, there's again been preclinical work, but unfortunately epigenetic modifying agents are a class of drugs that are emerging. It's a class of drugs that only in the last decade have drug companies begun to understand they need to develop, and the one that we're planning to bring to clinical trial for DIPG, panobinostat, is something that was just recently FDA approved for an adult cancer, but this is a very new class of drugs. And so I think that there will be a number of agents that are coming down the clinical development pipeline that hopefully will be efficacious in combination with DIPG, but we just are simply not there yet. And the drug that in preclinical models has shown some efficacy is not yet available for human use. I think that the issue, though, of drug delivery and the major issue of the blood-brain barrier is one that perhaps Mark could speak to. So I'll stop talking.
Thank you, Michelle. So two basic principles, and it's not unique to HDAC inhibitors or DIPG in treating central nervous system tumors in children or adults, has always been the delivery of the therapeutic agent. And getting that agent into the milieu or the parenchymal compartment of that tumor has always been a challenge. When you look at a fractional extraction of chemotherapy from the systemic circulation, it's usually measured in a fraction of a %. The answer to that has been to drive up the concentration to expect a higher proportion to get into the tumor at the expense of systemic morbidity and toxicity with bone marrow suppression and many other features with regard to the basic systemic functions of the human being. So those two counter forces have been very problematic in treating central nervous system tumors. And many years ago, inspired by some of the early work by Ed Oldfield and Russ Lonser at the NCI, looking at this idea of local delivery, I always felt in my heart of hearts that the DIPG is a very, very unique and really optimal tumor substrate to apply this toward. It's relatively small compared to other brain tumors. It doesn't necessarily metastasize or spread at the time of diagnosis. So really trying to lay that groundwork in something that really seemed inconceivable from the standpoint of putting small catheters directly into the tissue itself and infuse and perfuse that tumor with the agent has been highly appealing. So we've taken that from the laboratory effort over 15 years ago, and we're 3 years into our first phase 1 clinical trial doing this in children with a different molecule, not an HDAC inhibitor. But really the proof of principle has been superb. The toxicity profile has been extremely well tolerated. The fact that we can show with very good evidence 1,000, 1,500 times higher concentrations of drug in the tumor relative to the systemic circulation has been really profound. So the proof of principle, the feasibility, the safety, are soon to be finished. And I think with that, then we open up the doors to some of the talented individuals on the panel and beyond to say, well, if we want to optimize this, what should we optimize with now that we have proven we can do this in a very effective and safe way?
Great, thank you. So we are going to try, we have some more questions coming in. This is another long one that I'll try to summarize. And I'd say sort of the gist of this one from Jenny Mosier is, what are sort of the legislative regulatory obstacles to the research and the clinical treatments that you're pursuing? And what can the patient advocates do to help overcome that? Obviously, there is advocacy on behalf of the scientists themselves, just in terms of research funding and regulations, but what can the patient advocates and the family groups bring to this debate? Don't all raise your hands at once.
So the obstacle for research is, money is one of the obstacles, but it's not a major obstacle. I think we have now more and more funding. It's getting the right models, and it's really hitting the right. It's a completely new concept of tumor genesis, and this really has to be thought about from the get-go in order to have anything that's meaningful. And I emphasize it's not having material from patients, not only at diagnosis, but at specific time, at time where they relapse or escape a specific given therapy, is going to be very helpful to understand what is happening and have a more of a longitudinal and meaningful. So this is one obstacle, is convincing people of the need to do serial biopsy in patients where the life expectancy is very low. For parents of patients that died, they know about all of the heartache it is to just decide, do I need to put my child through this yet and yet again? And unfortunately, that's potentially one of the only way, if not the only way, for us to better understand this disease longitudinally. The number 2 obstacle is having pharma agree to combination of targeted drugs. If they haven't, so this is something that the people that we're being helped, but we need even more push on those pharma. In the US, people are lucky. I am in Canada, where there is 0 clinical trial that is open, and the pharma, just because we have another regulatory agency, which is Health Canada, opening up the door for treatments is a no way. For example, there is a clinical trial that is ongoing at the PBTC for nivolumab. I don't want to name Asians, something. We don't even know if it is effective, but there is a clinical trial, and you could potentially get answers from that. This drug, if the parents have to pay it in order for me to get something similar here, and each injection costs $5,000, not to mention the hospital costs and whatever, and this is one of the nicest drugs in terms of cost. Getting another oral drug, it is $100,000 just for a month of therapy or 2 months of therapy. So it's if you are part of a clinical trial, yes, and they are really open only in the US. The moment you go outside of the US, people are crying and whatever. We try and enroll, but it's for every country there are challenges, and clinical pharma are not so willing to open clinical trials for pediatric patients for orphan disease, except in limited number of centers. So I don't know. I took a long answer for a short question, but this is my view.
Denise, what do you think? I mean, what else can groups like yours be doing?
I sometimes feel like I just regurgitate the same thing over and over again. I feel like I say, we need more money, we need more awareness, doctors need to have more at their disposal to work with, because this tumor kills too quickly. We had 9 months with Caitlin from the time she was diagnosed until the time she died. And we tried hard. My husband is a family practice physician. I worked with the inpatient oncology population at a pediatric hospital in Orlando. Neither of us had ever heard of this tumor, ever. If you look on the NIH's website, if you look on the CDC's website, and you search DIPG or diffuse intrinsic pontine glioma, you will get 0 results in that search. As a parent, that's unacceptable that institutions that big don't recognize that diagnosis. So I'll keep talking, and I'll keep trying to raise money, and I'll do what I can in my little town with the people that continue to support us. And I hope that then other parents will do the same thing, and then maybe hopefully all together we'll be able to make a difference so that this phenomenal group of physicians can find a way to treat it.
Great. Thank you, Denise.
Does anybody else have anything to add to that?
No? All right. Well, we have another question from the audience, and this is a more sort of research-based question. So the hot topic in molecular biology these days is CRISPR. Do any of you on the research side see CRISPR playing a role in any of the DIPG studies, any of the clinical treatments, anything like that?
So yes, it's the short. So we're using CRISPR. A lot of it. CRISPR, what does it do? It's just either what it does very well is knockout, meaning it helps you remove specifically the expression of a gene. Okay, we're trying to do this, but we're also trying to add the mutation, which is a little bit harder using CRISPR, just to model it and to see what is it. So yes, it has a role, and to understand what is the role of the specific associated mutation with this histone mutant. So it's one of the tools that we're using. It's not the tool, the magic tool, but it's one of the many tools that we're lucky to have at our disposal with the next generation sequencing and the other advances in technology that those patients are benefiting from. And our drug screening, that Michelle can speak more about.
Great.
I think we have one more comment from the audience. I think I will. Oh yeah. It's Jessie Schumacher again. She says, I'm glad that Nada mentioned serial biopsy. For those patients that did not receive a biopsy at the time of diagnosis, is it clinically beneficial to that child to receive a biopsy during the honeymoon period following radiation? Is there any doubt if there will be any actionable findings? And it goes on, but we can add on to that.
When's the best time to biopsy?
Yeah.
I don't know when it's a diagnosis for fact. Why? Because it tells you what it is before radiation by itself induces other damage, other mutation, other things. It helps by time, but it adds on top of it. I don't know if it is at a time where tumor is shrinking, things are happy, patient is stable, it's the best time. I would do it at a time when something starts to regrow, or you have a solid argument that the tumor is regrowing. Mark, Kieran, and Michelle, what would be your input?
Yeah, I think the problem here is that there isn't a single answer. Obviously, the ideal in terms of the research and the understanding would be at the time of diagnosis, and then maybe when things have quieted down so that you could see what has changed, and then obviously when things get going again so that you can understand how the tumor has adapted to the therapy, and then as you apply more therapy to see how it changes. Now obviously that's easy when you've got a skin tumor or leukemia, for example, that you can just get a blood sample from. It's not really practical today to do multiple biopsies. And as we learn, since remember, we've just started, biopsying is a brand new thing for DIPG. The first time around, it's not like any of us have been biopsying them over and over and over again. As we learn, we'll certainly understand that. Obviously, the circumstances of the drug you're on, the way the biopsy can be done, whether there are any toxicities from the tumor, the radiation, or the treatment you've been on, like bleeding, those kinds of things that would increase the risk of a biopsy, those are the things that are also going to have to come into play. And then the other thing that Mark Swadane had mentioned previously is, unfortunately, as people are hearing that biopsy is possible, suddenly every surgeon, every small place now assumes that that's the standard and they ought to do it. And it's absolutely critical for parents to understand that as he previously pointed out, there's a certain level of expertise that is absolutely required, not just in terms of doing the biopsy. We get calls from surgeons who said, okay, we had a kid with a DIPG, we biopsied them, we've got the stuff sitting on the table, what do we do with it? And it's like, if you didn't know how to process it the minute you took it out, you've already lost most of the advantage of doing it. And it's this kind of coordination at every step of the way that's as important as the actual doing the biopsy itself.
This is why the biopsy should be done as part of a trial. Who really knows what is it that we're going to do with each sample, and actually if they're done in smaller centers, how to process the sample to get meaningful information from something that's potentially dangerous for the kid.
Yeah, I absolutely agree with what's been said, and I think it speaks to this larger issue of the tumor, you know, as all tumors do, evolving over time, and our need to understand how it's evolving, how it's responding to therapy, and how we can attack it at, you know, stage 2 when it comes back.
So we have another question from the audience. This is from Sandra Perkins, and I think she's getting to something that's actually a big question in the field. She says, her son was 19 when diagnosed and died 9 months later from DIPG. Is there anything different about DIPG in young children versus older children? And is this an area that's beneficial to explore?
Definitely yes. So this mutation seems to be present in the midline of the tumor, and there's 2 studies that actually included older patients with DIPG, 18 and above. There is more IDH mutation, which is another gene that's found mutated in high-grade glioma, and there may be other things. So there may be some variation, either in the histone mutation or in the partners, and potentially in the type of therapy. So at any age, the biopsy should be made, and we will learn more by also comparing what's happening in younger to mid. Already we know that there are two different histones that are mutated at the same residue based on age. One in the very younger and has other association than the others, and there may be something else in the older patient population.
And I think this speaks to the age differences and the different genomic, you know, phenotypes that Nada and others' work has uncovered in very young patients, you know, in the 3 to 4 year old range versus 6 versus older patients in their teens, speaks to the developmental origins of this tumor and our need to understand better exactly what is going on in postnatal brainstem development that is allowing for or promoting the development of these tumors, and why there is a different genetic signature, or slightly different genetic signature at various ages. It's a very interesting set of clues. We just don't yet know all of the answers.
Yeah, I would add to that not only in terms of age, where in some of the mutations we're seeing much more commonly in girls than we are in boys, suggesting that it may be even more complex. So by the time you take age as well as the mutation as well as the sex into account, we're beginning to see different patterns. And this kind of gets back to, I think, some of the themes, i.e. when to biopsy, how often to biopsy, what you get out of it. DIPG isn't a disease. It's a variety of nuanced differences that appear to use different mutations or different abnormalities to drive the tumor in different circumstances, which means there may not be a treatment. There may need to be different treatments based on the circumstances of what got your tumor started, what keeps it going, and only when you understand those variables do you come up with the right mix excuse me, of the kind of approaches that would be successful for an individual child.
So that actually leads to an interesting question, which the research around cancer in general these days is pointing to what you just pointed out, Mark, which is it isn't just one disease. There are all these different diseases depending on what the origin of the cancer is, where it metastasizes to. So is this something that we can bring in research and approaches that have been applied to other cancers to DIPG, or can DIPG inform the cancer field at large? Or is this something where it's going to continue on its sort of separate track, differentiated from, as you've mentioned, the major cancers that have gotten most of the attention up to this point?
I mean, I think the clear answer is both of the above, that we're not so smart that we can afford not to learn from the overlap of similar situations with other diseases. And certainly there are some mutations in DIPG that are also found in other brain tumors, in other adult tumors, and in other cancers outside of the brain. And obviously, you know, we often use the analogy of cancer being a little bit like a car out of control. And there are only a certain ways to do that. All of the mutations in the fender or the antenna is not going to drive it out of control. Whereas if you disengage the brake, if you stick the gas pedal, those are very good ways of making something out of control. And many cancers use the same mechanisms over and over. And our goal is to understand those. At the same time, there is a lot of variability. Again, it gets us back to this concept of DIPG isn't a single disease. And as we understand for any individual child's tumor, what made their tumor go awry, we're more likely to understand that. And if the breast cancer group has already found a drug that targets that approach, there's no reason we ought not to bring it in. And as Michelle already mentioned in some of her preclinical work, she's already found some drugs that have been approved for other adult cancers that are now being drawn into the DIPG field exactly because we recognize those patterns.
And I'll just follow on the beautiful car analogy, which I really like. If the brakes are in a different place in different cars and different cancers, the fuel, the gas, may be common. And so what we're learning about the tumor microenvironment for other brain tumors is turning out to be important and valuable for DIPG. And what we're finding out about the DIPG tumor microenvironment that promotes growth is similarly becoming relevant for other kinds of brain cancers. So there's definitely DIPG specific information that's important for other types of cancer, and there's definitely clues that we are gleaning from other types of cancer to understand how to fight not only putting on the brakes within this particular type of tumor cell, but also to cut the fuel to the car.
So we're in the last few minutes of this hangout. We've already asked our panel, our audience to send in any last-minute questions. But I just want to ask the panelists if there's anything we haven't touched upon during our discussion that you think is an important point to make.
Or any final words of wisdom or thoughts or anything like that.
I think one thing that's very dear to me and the way I approached DIPG was pediatric cancer are not adult cancers. They have really nothing to do. There may be drugs that will be working on specific subgroups within both that would be applicable, whatever, but it's really pediatric cancer, including DIPG, have nothing to do with the rest of cancers. And what is even more unique about this cancer, it seems to be very faithful in that it does include the original mutations. And the couple that you find it beginning, mid, and end. I'm re-emphasizing this because it's telling us something. This is where potentially the answer will lie in how to treat those patients. We're not going fast enough. I see patients, as all of my other colleagues, dying almost on a weekly basis from this. Even if it's a rare cancer, we see them in our centers, and nothing is soon enough. However, it may take a little bit of time because we need the right models, and then we'll make sense of what are the meaningful targets and how it is we can treat them.
Yeah, I would certainly reinforce that. I think the reality unfortunately for families that are experiencing this is we've made more progress in the last 5 years than we did in the prior 50. But at the same time, we still have an enormous way to go. If cancer were easily beat, it would have been done a long time ago. Remember that cancer has nothing to do all day but figure out how to get around what we're doing. And as soon as we do something, treatment A, they're going to go to plan B. And as soon as we introduce B, they're going to figure out plan C. They're very effective at doing this. And the sense of it that it's going to be easy, or simply because we've now biopsied them or found a gene that's mutated, that we're suddenly going to cure these tumors. Remember, we found many of the lung cancer genes 30 years ago, and for the most part, the only way to really not get lung cancer is to not smoke. We haven't been all that effective because every time we do it, the tumor simply develops a mechanism that gets around what we're doing, and there's no reason to think DIPGs aren't going to try the same thing. That question of should we be biopsying a lot, it would be ideal if we could understand how they adapt so that we could build into our therapies the anti-adaption approach. But I just think people have to realize you can't be in this for the short term. This is going to be a tough, hard, drawn-out battle. I think there's no question we are going to be successful. but it's going to take some time. And as I said, now that we understand that this is a disease that differs a little bit between patients, we may cure some subtypes before we cure all of them, but every time we make an advance, we get to move it forward for everybody.
And I would just echo exactly those sentiments. The more we study, the more questions are arising. I mean, this is an incredibly tricky, insidious tumor, and the more we understand, the more it's clear that we need to know. I think my hope is that the coming years we'll see some step forward, but by a step forward, I don't think we're anywhere close to curing this disease. I'm hopeful that we'll find a treatment that shows even a bit of efficacy. We still haven't done that in over 200 clinical trials. So I'm hopeful that that will come with the new insights that have emerged in the last few years.
And Keith Deserich of The Cure Starts Now, since he's on the event page, he writes his thoughts, which is that he thinks there's been some great research going on in DIPG, but they basically hinge on 4 main things, being data collecting data in tissue, the delivery of drugs, translational development, and clinical treatments that take from other cancers. But previously, DIPG seemed to have one focus mainly, and that was the clinical use of drugs that were piggybacked from other types of cancers. And now he feels that through collaborations, people can tackle all 4 areas. And we have one more question. It's actually, we've had 2 questions from the audience about immunotherapy as a form of treatment in DIPG. If you could hold the previous question, I think that it's basically immunotherapy has been getting a lot of attention in treating aggressive cancers. What are the panelists' thoughts on using something like immunotherapy? Does it do more harm than good?
Michelle, you want to go about this or Mark? There's been trial.
I wouldn't say that. I wouldn't say that immunotherapy is a very exciting opportunity, but I don't think any of us think, just like all of the other research, that it's going to cure everybody instantly or easily. It has a lot of toxicity with it because you're basically teaching the immune system how to attack your own body, which often can result in autoimmunity and all kinds of associated problems. But at the same time, it's had some successes in some diseases, and like everything else you've heard about today, we need to learn as much as we can from that expertise and see if and how we can apply it, on the assumption that it's not just delivery that's going to cure it, it's not just a drug that's going to cure it, it's not just radiation. If we can put all of these things together, potentially including immunotherapy, is where we're more likely to be effective in the long term.
Totally agree.
Great.
Yeah, and Denise, do you have any final last thoughts before we sign off?
It's an honor to sit on a panel with these incredible people who have dedicated their efforts and energies to a very unattractive disease. And on behalf of parents who don't yet know that they'll have to say thank you, I want to thank each of you for all that you're doing.
Thank you. So we want to say thank all of the parents and groups who are watching. We want to say thank you to all of our panelists and all the scientists who are watching and have participated. I think this has been a great discussion and hopefully sort of only the beginning rather than one-off. We do these hangouts through ASBMB frequently, so tune into our website, asbmb.org, to see when the next one's going to come up. Anything else from us?
But no, we just want to thank all our panelists for making the time and sharing their stories, and thank the audience for making the time to be on this hangout. If there were any questions that we weren't able to get to, we will go through the page and send them to our panelists to answer offline if there are any questions that we didn't get through during this broadcast. But thank you all.
And this will be posted on our YouTube page. If you missed any of it, it'll be there for eternity, so you'll all be on there. All right. Thank you, everybody, for watching. Bye-bye.